Okay, let's go ahead and get started again. A couple of quick announcements. First one is the biology department seminar this week. Kiki Sanford, who's a blogger, science journalist, science communicator, is giving our seminar this week, just talking a little bit about science communication. If it's something that any of you are interested in, maybe career-wise, etc., highly recommend it. Also part of Darwin Day, that's this week, also on Friday. I think it's Friday. There'll be an evening seminar in Smith Center Ballroom. You can get free tickets over at the box office. The same thing is true for Eugene Koonen's evening talk a week from Thursday. But you can only pick up those tickets starting on the 11th when this seminar is. Anybody make it to the chemistry seminar last Friday? Methyl transferases. I wish you'd actually gotten to the stuff at the end, because I think that would have been much more interesting, but you know, that's my own personal bias as far as that's concerned. So today we're going to talk about mostly RNA processing, but beforehand we need to review, and review of course means clicker questions, what we talked about last time. So the most similar sequence in bacterial sigma 70 promoters, we'll get back to that in a second, to the eukaryotic BRE in terms of its location and function in eukaryotic promoters. So, okay, we're looking about the similar sequence in bacterial promoters, is the Tata box -10 sequence, -35 sequence, INR, or DPE. Ten, 5. Okay, so a couple of critical things about this question. The most similar sequence in bacterial promoters. So what does that rule out immediately? Tata boxes are in eukaryotic promoters. INRs, initiator sequences, are in eukaryotic promoters. Downstream promoter elements are in eukaryotic promoters. Okay, so we're left with -10 and -35, right? Yes? Makes sense? Good. Okay, so -10 is relative to where the transcription starts. -35 is going to be further upstream than that. Where is the BRE relative to the other promoter elements, like the Tata box? Upstream of the Tata box, and also really important for determining the orientation of transcription, because a Tata box is the same whatever strand it's on. So I guess I did not do a very good job of explaining this, because most of you liked A. Tata boxes are not present in bacteria. So, yes?
When you bring up the boxes, can you just move it farther to the side if people want to copy?
Yeah, I can. So when here, I moved all the numbers. Hang on, let me move this here. Over to this side so that people could actually see them. So that's why it's a little overlapping here. So people who are on this side can actually see them. The problem is partly this room, to be perfectly honest. So yeah, it's not Tata boxes because Tata boxes are specific for eukaryotes. And I guess I didn't do a good job of mentioning that. The -10 sequence has lots of A's and T's in it, but it's not the same as a Tata box. So maybe that's partly where some of the confusion has come in. So and the orientation, as far as promoters are concerned, orientation in promoters, because we talked about last time, you can use one strand or you can use the other strand in terms of transcription. It's going to depend on where you get the polymerase bound and what strand it's going to be bound to. And that's going to depend on your -10, -35 in bacteria, which is where the sigma factor binds. And one of the things that I haven't talked about yet, but we'll talk about more later, is that there are multiple sigma factors that you find in bacteria, not just one. We've only talked about one so far, but to be correct in the question, that's why I put sigma 70 in there. So where that sigma factor is going to bind is going to determine which strand you use, and then, of course, which gene ends up being transcribed. BRE is what? It's the TF2B recognition element. It means it's something in the DNA, any element of something in the DNA that TF2B is going to interact with. Because it's upstream of the Tata box that gives you an orientation, tells you what is downstream.
You said what interacts with it?
The TF2B complex interacts with it. Okay, does that make more sense now? Yes, no? Yes?
And I feel like I'm the one that's always asking about the upstream, downstream. And the upstream is towards 5?
Okay, so the question has to do with upstream and downstream. I had a question on D2L, which unfortunately I didn't have a chance to answer about this. So upstream and downstream, and people ask me about this after lecture too, so it's a good thing you're not the only one who's interested in this. So all of that depends on where the first nucleotide is, where the first nucleotide is in your chain that the RNA polymerase makes, and relative to where that nucleotide is, it's going to have a 5' triphosphate and a 3' OH. And so anything relative to that particular first nucleotide, which is 5' to it, will be upstream. And relative to the 3' OH of that nucleotide is going to be downstream. But you've got a template strand and a non-template strand, which are going to be opposite orientations. So you can't really say which one is 5' versus 3' unless you talk about template versus non-template strand. So base it on the first nucleotide and the transcript, which is being made, which is the RNA. Okay, other questions on this now? Okay, so our second clicker question of the day, it's a little bit further along in the lecture. Which of the following bind directly to specific DNA sequences? TF2B, TF2D, TF2H, or Stedman's favorites, A and B, or A, B, and C? Just because there's favorites don't necessarily mean that they're right. Basically means he's lazy and he can't think up of any other answers. Ten. 5. Okay, there seems to be a lot more consensus on this one, which is nice. So we'll show the results. Is that okay in terms of where it's oriented? You can see everything else here. Maybe move it down a little bit. So hopefully, last discussion made it really clear that TF2B binds to the BRE. So certainly everyone should get B here. But then a lot of people think D because it's A and B. Why do people think that TF2D binds specifically to DNA? What does it bind to? Binds to DRNA. And actually it doesn't bind to the BRE. TF2D binds to what? Okay, the downstream promoter element, yes, and the RNR, and what else is left? The Tata box. So TBP is part of TF2D. And I would expect you to remember that because I thought we had talked about it a lot last time. So for those of you who didn't get that, maybe I wonder if it always gives me like the most answers to the first one here. Curious. Okay, so I did want to put up a couple of things here that are much more up to date than we have already in the textbook. Last time we talked about what TF2H does. It's a helicase because it's got that H, right? Well, actually, it's nothing to do with it, but it's an easy way to remember it. And has a kinase activity. That kinase activity phosphorylates a particular part of the RNA polymerase. The particular part is called the CTD, the C-terminal domain. And nobody knew what the C-terminal domain actually looked like until we had a structure for this. And this is literally from last year. In fact, published a little before it came out in print. The main thing here is not so much where the CTD is, but it's to give you a bit of an idea of how ridiculously complicated this whole structure is. Here is what it looks like. It's kind of a blob, but mostly to show that you've got TF2B, TBP, TF2E, TF2F, TF2H, the whole RNA polymerase, etc. This thing is humongous when it's sitting down on one particular promoter. And just for scale, again, the DNA is right here, and you can sort of see the DNA bent through here. So the amount of proteins, the amount of everything else which is there, is vastly outweighing the DNA. So it's a really large amount of protein complexes. And all this is about is finding the right place to start, which should also give you an indication this is a really important thing to do, which again is not surprising because if you look at the whole genome, most of the genes are not turned on. And the reason is they don't have an RNA polymerase that's associated with them. And so this is just supposed to give you that bit of an idea here. And the CTD sort of hangs out in this area right next to TF2H, again, not surprisingly, because that's where you have phosphorylation that takes place. I wanted to spend a couple of minutes talking about transcription in Archaea because Archaea are some of my favorite organisms, but they're also a great model system for studying some really basic molecular biology in a eukaryotic-like system. And why is that, particularly for transcription? In Archaea, as we talked about briefly last time, there's only one DNA-dependent RNA polymerase instead of 3. But it still has about the same number of subunits, and the structure is practically identical between the archaeal RNA polymerase and RNA polymerase II. In eukaryotes, and RNA polymerase II transcribes what genes mostly? Messenger RNA genes. So nice model for looking at messenger RNAs. The archaeal system only requires 2 general transcription factors, so why doesn't Stedman not bother with all the other ones? Because that's what happens in the eukaryotes. There's just a TBP, so no TF2D complex, which is a whole bunch of proteins. It's just the Tata binding protein, and just TFB, and these are just TF, not TF2, because there's only one RNA polymerase. So TFB and TBP bind to the promoters. Then you get the RNA polymerase bound. You have a closed complex where both the strands are together. An open complex, you don't require an extra helicase for this. So it's a lot more like the bacterial RNA polymerase. And then you have a board of initiation, just like you would normally have in all the other polymerase systems. And then a change into this elongation complex. Another way of looking at this is in a cartoon form, what that eukaryotic promoter looks like, TF2H, D, etc. And we've got this nice simplified complex here with just TBP, which also bends DNA in a very similar way, TFB, and the RNA polymerase. So that's great, it's wonderful, it's a really nice, simple system for trying to understand some of the very basics of what's going on here without having to deal with all of this extra stuff. I won't call it junk. There are some slight modifications that happen and actually make the archaeal system a little bit more like the bacteria, and that is that some archaea have multiple TBPs and multiple TFBs. And so they can mix and match and exchange these out in terms of different promoters. And since we just talked about what the specific DNA binding properties are of TF2B and TF2D, since TFB and TBP are those equivalents, these guys are also bind to slightly different sequences. And so by having a different sequence and a different general transcription factor, you can direct the polymerase to a different promoter. And just to sort of beat the dead horse here, these are some X-ray crystal structures of the archaeal system, Pyrococcus furiosus, that Dr. Bartlett's lab here up on the 4th floor of SRTC works on, and the human system. This was published first, before this one. Yes, we beat them again. TFB and TBP associating with a piece of DNA. And other than the colors here, which are different, purple versus red and green versus yellow, these are practically identical. So they're really nice systems for studying the basis of what's going on at a mechanistic level for RNA polymerase II like transcription. So promoters for RNA polymerase II, you've got the sequences. Now this is DNA, TATA, and TF2B recognition element. In Archaea, you have a TATA box and a TFB recognition element. So extremely similar in terms of promoter sequences. The RNA polymerases are very similar to each other. RNA polymerase II in eukaryotes has 12 subunits. Archaea have between 10 and 12, and those structures are extremely similar to each other. However, there's only this C-terminal domain in eukaryotes. It's not terribly surprising because what did I tell you that Archaea don't have? They don't have a TF2H and no TF2H homologue. What does TF2H do? Helicase activity and phosphorylates to CTD. Well, okay, we don't have those in Archaea, and so you're not going to need them. What you need in terms of factors, and remember again, factors are proteins, whereas these promoters or elements are DNA sequences. In eukaryotes, you need TBP, which is part of TF2D, TF2B, and a whole bunch of other ones, whereas in Archaea, you just need these two. Chromatin, which we talked a lot about when we talked about chromosome structure, got nucleosomes, all kinds of nucleosome regulatory complexes, histone modification enzymes, chromatin remodeling complexes, etc. There are a few histone-like proteins in some of the Eukaryotic, which is one of the groups of Archaea, but not in all of them. So basically, archaeal transcription is the way to go in terms of studying transcription. So that's all I wanted to say about Archaea as far as this is concerned. Did want to take a quick look at this transcription movie here. You may need to reload it. Is it going to start again or not? Doesn't look like it. So here is a typical eukaryotic promoter, clearly missing all of the nucleosomes, and a couple of general transcription factors bound to it. This would be your TF2D complex here, bound to the TATA box, causing a nice little bend in your DNA. These guys are other parts of TF2D, which are just waiting to interact with some of the other parts of your transcription initiation complex. Most of the time, they'll just be sitting here. Now we've got the RNA polymerase, which has gotten associated with the DNA. We've got those extra factors. There's TF2H, all ready to go, but we actually haven't started yet. And we'll talk much more about this later on. Most cases, you need an activator, which comes in, interacts with your complex, and then the RNA polymerase undergoes this conformational change, becomes a highly processive polymerase separating the two strands, and making your nice little RNA up here in yellow at the top. Here, it's shown as shooting along the DNA. As we all remember from last time, it's considerably slower than any of the DNA polymerases because probably of just the way that this whole complex is working. But it's only one protein complex that's doing this. The elongating polymerase is just one thing, as opposed to a replication fork where you've got all the clamp loaders and the clamps and the primases, etc. Here it's just that one protein moving along the process. So let's move back to here. Yep. And any questions, more questions about transcription? Talk much more about transcriptional regulation later on. Yes?
So even though you just have what you were saying is just that one protein moving along, that RNA polymerase II activity is still slower than both bacterial and eukaryotic DNA polymerase activity?
That is correct. So the RNA polymerase is considerably slower than all of the DNA polymerases, at least the replicative DNA polymerases that we've talked about. Some of the repair polymerases are slower. But other questions on transcription? Otherwise, we'll talk about what happens to the transcripts once they get made. So again, overview of what we're going to talk about for the rest of today. Start out talking a little bit about splicing, capping, polyadenylation, RNA export, and then a little bit about, if we get there, how our ribosomal RNA gets made and how our ribosomal RNA gets modified, which is a very important process. So splicing, capping, polyadenylation, RNA export, all of this is just happening in eukaryotes. And that's because eukaryotes have this wonderful subcellular compartment, the nucleus, that they put all of their DNA into. In that DNA, you have your genes. Your genes are made up of introns and exons. Those exons are what's going to be making the protein. What happens, which is really rather inefficient, and we'll probably see why that inefficiency is okay later on, is you make what's called a primary transcript. And that primary transcript is actually an RNA which encodes all of the introns and all of the exons. And as hopefully we all remember from previously, those introns are usually way, way, way bigger than the exons. And so you end up making a much, much longer transcript. This is what RNA polymerase II does. And then you have all of this processing, capping, splicing, polyadenylation. All of this happens still inside the nucleus. Once all of this has happened, then you have export, and then this messenger RNA is translated into protein. So you go from a pretty big transcript down to your final transcript, and that actually gets made. Prokaryotes are way smarter. Well, maybe. But all of this is happening in one compartment. So your DNA is in one compartment, your transcription happens in one compartment, and your translation happens in one compartment. One of the things that this means is that the whole process of going from DNA to protein is much, much faster in prokaryotes because you're making these things, and actually you can translate a messenger RNA as it's being made. And it turns out that that's something which almost all, when we say here prokaryotes, mostly we mean bacteria. A lot less is known about Archaea, but the same thing seems to be happening there. So if we look at these RNAs, again, not terribly surprisingly, because these prokaryotic messenger RNAs are being made and being translated simultaneously, you have where you started, 5' triphosphate, your coding sequences. There are very few introns in bacterial genes, also true in archaeal genes, but usually when people talk about prokaryotic, and that's definitely true for this textbook, they mean just bacteria. You have triphosphates at one end and coding sequences with very few introns, and quite often, not all the time, but quite often in these bacterial messenger RNAs, you have multiple different proteins which are being coded for by one RNA. So in this one RNA is between where you start, it's nucleotide +1, and where that RNA is terminated here at the 3' end. having multiple different proteins, and this is completely different proteins relative to each other, is called polycistronic. And if you're interested in cistrons, we can talk about what a cistron means offline. But multiple different genes and completely different genes encoded for on one RNA is a polycistronic RNA. Of course, the opposite way is a monocistronic RNA. This is true for the vast majority of eukaryotic messenger RNAs. Only ends up coding for one protein. There are a couple of other differences. Again, hopefully none of this is new to you. There are cap structures which are present at the 5' end of most eukaryotic messenger RNAs, and poly-A tails. And these poly-A tails are not coded for in the genome. They're added after transcription takes place, post-transcriptionally. Poly-A tail, turns out that both of these are really important for mostly translation, but also for other RNA processing things that happen inside the nucleus. And then, of course, this protein which is made here. So let's start at the 5' end and move our way down towards the 3' end. 5' end has this cap structure. Cap structure is literally a single guanine residue which is put on in a 5' to 5' linkage at the very end of the messenger RNA. And the main reason for this seems to be protecting that end from the nasty exonucleases. Lots of exonucleases around. Exonucleases are going to be looking for a 5' phosphate to chew in on. Well, if you have a 5' bond, there's now no longer a 5' phosphate to actually interact with. And so that's one thing that's done. Also, this provides a very specific structure that other proteins can interact with. Those other proteins are called the cap binding complex that we'll see is really important for particularly getting that messenger RNA outside the nucleus and making sure that it gets translated properly. So how do you get this really bizarre structure? 5' to 5'. We've never seen 5' to 5' happen before. So how does that happen? First, you have your normal transcript, which is made. And the first thing that happens is one of the phosphates is taken off at the end. So you have the triphosphate. Why triphosphate? Because RNA polymerases are great. They can start a polynucleotide chain with a single nucleotide. And so they always have those triphosphates at the end. Take one off, and then you have one of my favoritely impossible to name enzymes, the guanylyl transferase, which takes a GTP and through our normal hydrolysis process, get rid of the pyrophosphate. That pyrophosphate gets broken down into individual phosphates, hooks up this, started as a GTP, as a GMP, now at the 5' end. So now you've got guanine, 3 phosphates, and then whatever the first nucleotide, this would be your +1 of your transcript. Then there are multiple methyltransferases. The first one methylates this guanine, and then other ones can or cannot, it depends on the particular cap, will methylate the ribose here at your 2' position. So if you have a 2' O-methyl ribose, so OCH3 that gets added onto here, could you do this with DNA? No, you couldn't, because there's no O there to put a methyl group onto. So you can't make these O-methyl riboses there. So now you've got methylated base, methylated guanine, and then some methylated riboses here. Cap 0, cap 1, and cap 2 just are relative to the number of riboses that have these O-methyl groups on them. So cap 0 has none, cap 1 has 1, always on this +1 position. Cap 2 is +1 and +2. So that's our cap structure. Again, cap binding complex will associate with this, pull it out of the nucleus, protect it from the nasty exonucleases, and help with translation later on. Okay, we're happy with caps? Okay, now we want to talk about what happens in the middle. To understand what happens in the middle, I do need to introduce the so-called R-loop technique. This is a purely in vitro in glass. Of course, nobody does glass anymore, so it'll all be in plastic. But it's a process which is not a real process. It's just a way of looking at your DNA and your RNA together. But it's an RNA that's already been made. It's an RNA that's got its cap, it's got its tail, etc. And so what you do, if you want to find out where on your piece of DNA that messenger RNA came from, is you take your DNA, you take your RNA, and you heat them up and cool them back down. And because the wonderful aspect of DNA can pull itself apart, put it back itself together with the appropriate base pairs, if you do this properly, the messenger RNA will now base pair to the DNA, and you end up with the non-template strand, which forms this loop structure. So this is a case of literally what people have done. So the DNA is here. It's your double-stranded DNA. Here is where your RNA is base paired to that, and then a single-stranded DNA has been looped out, and you have this little end here. This end here is just single-stranded, in this case RNA. Here's a little single-stranded RNA. That's going to be your poly A tail because it doesn't pair anymore. And here, the 5' end actually turns out to be really, really interesting because it can now, doing exactly the same technique, sometimes base pair to other DNA fragments, which is really bizarre because, wait a minute, now we have our sequence, we had our RNA, and it's been transcribed from the DNA, but if you look at where it's going to base pair to, it can base pair to multiple different pieces of DNA. And this is how people discovered splicing in the first place, was that they looked at messenger RNAs, and they did this R-loop technique, and they bound it to DNAs, and they said, whoa, wait a minute, we've got all these bizarre loops that are forming here, and so the DNA is not base pairing to the RNA in all places, but there are these other bits and pieces which are base pairing to our final, again, already processed RNA. The classic example of this was by Phil Sharp. This is the cartoon image. If you look at the actual image in their paper, it's a bunch of black dots on a gray dot background. It's really kind of hard to see, so color-coded is a lot easier. But basically, it's another one of these R-loop kinds of things. And so the R-loop is this particular RNA here, which is here in the dark green with the black at this end. Here's the DNA before it's bound. Here's the DNA at the end after it's bound. And then there's these loop structures, which you see in your R-loop. Looped out DNA, looped out DNA, looped out DNA, and then all of the rest of it. Turns out that the only way to do this properly is to have lots and lots of one particular RNA. In this case, it was from a virus RNA. And for a long time, people thought that splicing only happened in viral RNAs. And then they figured out it happened in all RNAs. But the main reason for that is you can get a lot of virus. You can get a lot of virus RNA. And particularly, this is the RNA which codes for most of the cap protein. The other thing that you need to do is have very specific pieces of DNA that you're going to be interacting with. You have to know exactly how big those pieces are and where all of the genes are on that particular piece of DNA. And the way to do that was basically to have lots of restriction endonucleases, which probably means something to most of you. All the people will call them restriction enzymes. I'll talk more about those at the very end of the class in terms of what's going on with molecular biology techniques. Again, mutant viruses from hell. Take the class next term. It'll be great. But the two people who figured this out, Phil Sharp and Rich Roberts, both got the Nobel Prize for doing this. Both published in 1977, but both were working on viruses and restriction endonucleases. And in fact, Rich Roberts founded a company called New England Biolabs because he was getting fed up with sending all these restriction endonucleases to other people who kept asking for them. He said, well, you can buy them from my company now. But what happens here is that you have a pre-messenger RNA, or also people call this the primary transcript, which has everything. introns and exons. And then, together with about 5 RNA molecules, and it turns out a whole bunch of proteins as well, but just the RNAs are enough to do this, you can chop out all the yellow things and end up with all the red things. So fusing all of the exons together. What happens here is that all you're doing is forming and breaking phosphodiester bonds. And so it turns out that this is almost isoenergetic, because for each bond you break, you're forming a bond. And yes, there's going to be a little bit of heat involved, so it's not quite isoenergetic, but it's really, you don't need any extra ATP hydrolysis in terms of the actual splicing reaction. Where you do need ATP hydrolysis is getting the specificity. When I say specificity, that's exactly where you form any of the individual splices. And we'll take a look at this a little bit later on. Why is splicing probably so important? And we'll talk about this much more later on, is that not all exons get used in every gene. And so that's how you can have some evolutionary processes which are going on there. So how does most splicing take place? When I say most, you should be thinking, oh, that means there are probably some other ways too. Yes, there are other ways that happen as well. But for the vast majority of our normal messenger RNAs, splicing takes place like this. So you've got an exon sequence here in blue, an intron sequence in yellow, and in that intron sequence is a very specific adenine residue, but the adenine itself is not as important as the 2' OH, which is in that adenine. 2' OH, the ribose of that adenine, then will chop the phosphodiester backbone here, and instead of just chopping it, letting it flap around, it actually will form a covalent bond to this adenine, makes this nice little loop structure. Then this 3' OH here can go and exchange with the phosphate at this end, and you end up looping these two guys together. What does this structure look like when we zoom into it? Here's that adenine residue, which is in the intron sequence. The 2' OH forms a 2' 5' phosphodiester bond. So we've already had 5', 5', 5', 2'. That's why I keep beating on 5' and 3'. It's important in terms of getting these things. Makes this big loop structure, and then this loop ends up being removed, and that's in fact how people figured out the mechanism here, is they were able to detect this loop structure and see how it was formed. What do those sequences look like? We talked about consensus sequences last time, even the time before, where you line up lots of sequences where we talked about promoters, and the most common nucleotides you find at particular positions, those are going to be the ones that you have most of your interactions happening with. Well, if you have a consensus sequence for splicing, the problem is that you have a very poorly conserved set of sequences which are there. And basically, you think about RNAs, you're going to have these sequences, AGGU and CAGG, very, very frequently in your genome. And this is partly why, when we started out lecture last time, I mentioned that predicting where splicing happens is very, very difficult. And that's because it's very difficult to identify specific sequences in all of the other stuff which is in there. So it turns out that detecting splicing is very, very difficult. I mentioned before there are 5 RNAs which are involved in doing this. These are slightly further down in that table that we talked about last time. These are the SN RNAs. So SN stands for small nuclear RNAs. Well, yeah, splicing is all happening in the nucleus. U1, U2, U4, U5, and U6. We will get back to U3 later. Don't worry. But for the time being, it's just these U1, U2, U4, U5, and U6, which are absolutely required for splicing. There's a nice animation here. I just added it down here at the bottom. We won't take a look at it right now. So what happens with these specific RNAs which are required for splicing? Not surprisingly, these RNAs have specific sequences, And those specific sequences, also not surprising, you're going to base pair right where you're going to have all the splicing taking place. So what things do you need to know about your splicing? You have to have your 5' end of your intron, your 3' end of your intron, and where that A is in the middle, where you're going to form the 2' to 5' phosphodiester bond. How are those found? How do you identify them? Almost always through these interactions with the RNAs. So here, RNAs, just to confuse you, they've done a typo here. So it's not snRNA, but snRNP. And the reason it's called an snRNP is because, as well as the RNA, which is in green here, we also have the protein in gray. So they're ribonucleoproteins. So snRNPs, which are here. Of those snRNPs, anything which is in red here, these are the important. Oh, why would I say important? Particular snRNAs. And then we've got a couple of proteins as well that are important here. The BBP, again, as if you didn't have enough acronyms to remember already. This is the branch binding protein. Branch is where you're going to have that lariat structure that forms, the looped structure to that particular A here. You also have the U2AF. Whenever you see an F, what are you going to think about? F is factor is going to be a factor, exactly. And factors are what? Proteins, exactly. So here we've got a U2, and in terms of that, it's an activating factor. So it's going to interact with the U2 snRNA as an activating factor. And it turns out that binds to the branch binding protein, but also is very close to the 3' splice site. But just because this U2AF interacts with the BBP, not surprising, U2 is also going to interact with this branch point. So BBP, U2AF, U2, U1 is going to base pair here at the beginning of your intron, also known as the 5' splice site. So the 5' splice site, the 5' end of the intron, is going to be the 3' end of your exon, because it's just cutting in that phosphodiester there. Then you have the association of this complex, U4, U5, U6. I'll bring up the latest picture of it on lecture on Wednesday. Turns out one was published last week. So this U4, U5, U6, this is really the sort of active amount of your whole process. All this process is also called the spliceosome. Again, more zomes. Spliceosome, any zome is usually going to be nucleic acid plus protein. So here, your snRNAs plus the proteins are going to be the spliceosome. Because you've got all of these RNAs, which are going to be in base pairing interactions with each other, turns out that the base pairing, and particularly the unpairing of those bases, is the most critical part in terms of making sure that splicing happens properly. And so what kind of enzyme do you need to pull two strands apart? A helicase. What are these things here? What is your messenger? It's an RNA. What are the snRNAs? These are an RNA helicase. So the RNA helicases are really important for pulling those base pairs apart. Why do you need to pull the base pairs apart? That's basically what's shown here. So U1 is going to bind at the 5' end of your intron. So here it is. The actual base pairs here are not important. That's why I'm not mentioning them. So binds here at the 5' end of the intron, and then you have RNA helicase activity. Now instead of binding to U1, you're binding to U6. Branch binding protein, also rearrangements take place here. This is not an RNA helicase because this is not an RNA, it's a binding protein. Now has U2 that's bound here. Really important with U2 is all these base pairs are great, and it sticks this A out. And that A gets pushed out so that the 2' OH is going to be available to make that 2' to 5' linkage. After you have this interaction with U2, there's yet another, now RNA helicase activity, which changes how U2 is binding to U6, how U5 comes in. Again, it's 4, 5, 6 together, which lead to this ligation of your exon to your exon and the removal of this intron structure. So it's all about RNAs getting pulled apart, put back together, pulled apart, put back together, pulled apart, put back together. And all of those interactions, anytime you have an OH, and it's the OH that you're starting out with, it's on the OH of that adenine in the middle. That's what starts the whole reaction. So that will come in and first break the phosphodiester backbone here at the 5' end of the intron, and then this 3' OH can now come in and bind to the 5' phosphate at the 5' end of the next exon. And so that's how you get this whole process. What does this thing looks like? This is the spliceosome in a cryo EM model. This is actually just the snRNPs without the messenger RNA. People think the messenger RNA kind of goes through here. But the message again here is that you've got this big complex doing something on a relatively small nucleic acid. So we looked at transcriptional initiation, small DNA, big process here, spliceosome, lots of stuff together, it's RNA and protein, coming together to make these kinds of interactions. One of the big questions that came up when people saw these consensus sequences that were not very well conserved was why aren't there way more mistakes that happen? Why does splicing happen so well? And the answer to that, there are a couple of answers for it. One is that splicing happens while the RNA is actually being made. So the RNA is being made from 5' to 3' by RNA polymerase II. As that first exon comes out, you'll start to assemble U1 on it. As you continue through the first intron, now you'll have the branch binding protein and U2 associate with that. And so it's going to be that first exon is going to get cut out before you've made all of the other exons, introns, etc. So you're going to be splicing while the RNA is being made. Usually, whenever people will cartoon these, you have your primary transcript, and then it gets spliced, and then you have all the tailing, etc. that happens. All this stuff happens simultaneously. So you have transcription, splicing, and then tailing. All of that then sends everything back out. So it's a co-transcriptional process. The other thing is that exons are recognized by specific protein, actually not specific protein, pretty unspecific proteins actually, which just bind to exons. And these exon binding proteins are very confusingly named SR proteins. Why are they called SR proteins? Because they've got lots of serines and arginines in them. So these guys bind to introns, and not surprisingly, they're also proteins that bind to exons. So these guys bind to exons, SR proteins bind to exons, and other proteins bind to your introns. Again, this is not my decision in terms of how we use a name. These are RNPs, so ribonucleoproteins, nuclear, and H just stands for heterogeneous, so a whole mixture of different proteins that are going to be binding to your introns. So the really critical part of what introns get used, get cut out, what exons end up in your final messenger RNA has to do with binding of U2. Why binding of U2? What is U2 binding to? Forgotten, that was like 5 minutes ago. Monday morning, my caffeine hasn't kicked in. That's what's giving you that A, the A with the 2' OH, which starts the whole thing. So it's going to be binding of U2, which is the critical determinant of what introns get cut out, and by corollary, what exons actually end up getting used. How do you get U2 there? You have U2AF. So the binding of U2AF is really the most important part about deciding which exons end up getting used. And this is just one example down here at the bottom of what can happen if U2 interacts with the wrong place or with a different place. So here I have a simplified primary transcript with 3 exons in it. If you had normal splicing, all the exons used, you'd have exon 1, exon 2, and exon 3. U2 would bind here right at the beginning of exon 2. You'd end up with exon 1, 2, and 3. If, however, U2 doesn't bind here, then you end up just going to the next exon. In many cases, it's not just the exons. You'll have different splice sites, sometimes even inside exons. And what this means is you've got a huge variety of what can happen. You've got a primary transcript, but that can get chopped and religated in lots of different ways. Yeah?
I think I'm having trouble understanding how U2 decides which exon it wants to use.
Yeah. So we'll see in just a second there. It has to do with interactions with these SR proteins. So again, it's that binding which is most important, but that binding is really starting the cascade. And so it's getting that into the right place. And again, most of that has to do with the interaction with these SR proteins. So talked a little bit about this before when we talked about the genome. Most exons are about 150 nucleotides long. 150 nucleotides, 3 nucleotides per codon in the genetic code, about 50 amino acids. About 50 amino acids is sort of the standard size of a domain. So most of these exons are going to be encoding for a domain. So if you're mixing and matching exons, what does that mean about domains? You're going to be mixing and matching domains. Each domain has what? Particular structure. And structures have function. Exactly. So mixing and matching exons is going to give you mixing and matching structures. This is not a very important thing down here. We talked about this before, but exons are almost always about the same size, very well conserved. Doesn't matter what organism you look at. Introns, on the other hand, are all over the place, and particularly in humans versus worms and flies, our genome is a lot bigger. Not surprisingly, our introns are also considerably bigger. As I mentioned before, most splicing takes place with U1, U2, U4, U5, and U6, but there are other splicing processes which take place. The numbers here are not important, but it all has to do with the specificity. Where do you start where you're going to be cutting out your intron, and where do you have the binding where that intron is going to be getting chopped out? The actual functional process, U4, U5, U6, is identical for all of these guys. So our normal splicing process is going to have U4, U5, U6. These alternative splicing processes also all have U4, U5, U6. And so the important message here is it all depends on where you find to start your specific intron with and where you have that A that bulges out. Very few cases you can actually take 2 different RNAs and hook them together. Really cool process, but also means it's even more confusing in terms of how all of these things put together. Because of this kind of so-called alternative splicing, either having different snRNAs, which are going to give you different splice sites, again, depending on where your SR proteins are, where you have the association with U2 with the RNA, you can use various different exons, and those different exons are going to give you different function. And what's best understood in terms of human cells is very often where what particular cell types this kind of splicing is happening in. So this particular example, the alpha-tropomyosin gene, multiple exons, multiple introns, just depending on what particular cell type, tissue type you're in, you're going to have different splicing, which is going to include different, usually now domains, which are going to give you different functions. So turns out tropomyosin in fibroblasts, you need something different than what you need in brain, or different in various different muscles. About 60% of human genes are known to be differentially spliced. So 40% aren't, 60% are, and usually it's just a couple of differences. It turns out tropomyosin is one of the more extensively alternatively spliced genes. We can only predict about 80% of these just by looking at sequence and looking at sequence conservation. So this is what happens in humans. Turns out that other organisms, and particularly the fruit fly, go really nuts as far as alternative splicing is concerned. This is one gene in Drosophila, DSCAM1, which can generate 38,016 different splice variants, and probably actually does generate these 38,016. Has to do with the nervous system and its development in Drosophila. That so much is not important. But what happens here in this case is that most of the exons are used. And that's true also here. If you look at tropomyosin in humans, most of these exons are in most of these final transcripts which are made. It's only a couple of exons which are going to be different. Same thing is true in DSCAM1. These first 3 exons are always used. It's exon 4, there's 12 different possibilities for. Exon 6 has 48, exon 9 has 33, exon 17 has 2, and I give you one guess what 12 times 48 times 33 times 2 is. 38,016. So those are all the different possibilities that you get in this particular process. So mentioned, hopefully again, beating the dead horse here, splicing happens co-transcriptionally. To some extent, what splicing introns and exons get used depends on how fast you're transcribing. So last time I mentioned, that the polymerase goes 20 nucleotides per second. Well, that's on average. Turns out that the RNA polymerase, unlike in that animation where it's sort of zooming along the RNA, which clearly has no histones associated with it, which is clearly incorrect, it's going to depend a lot on what different either proteins are bound to your DNA, but also in terms of some of the sequences that are present in the DNA, which is going to determine how fast your polymerase is going. So it turns out if your polymerase is going very slowly, it's going to be making the messenger RNA relatively slowly. You're only going to have one exon which has just come out, the next intron comes out, the next exon is there. You're going to splice between those two because you don't have another exon to splice to. However, if your transcription is going really fast, say in a region of the genome where you don't have nucleosomes, or you have very few nucleosomes, they're not compacted, then you may have multiple exons which are in your transcript, which the splicing machinery can choose between. And a lot of this also has to do with the modification of the histones, again, which is binding more tightly to the DNA, making it harder to transcribe, binding less tightly to the RNA and making it easier to transcribe. So getting back to your question about how do you get U2 associated here, it's really about these, now they call them RS proteins rather than SR proteins. Again, it's just because they've got arginines and serines in them. So the interaction of these SR proteins with your exon, these interact with U2AF, which is going to stimulate U2 binding. That's going to cause your exon to be used. If, however, you have no binding of your SR proteins, the names of these proteins is not important, then no binding of SRs, you're not going to stimulate U2AF, you'll go to the next exon. This is a very simplified process of how that's happening. Another way of looking at this, again, the main point here is that you can mix and match lots of different things in your RNA. So, okay, the whole thing, beating on only 1.5% of our genome is coding for specific proteins. But depending on which of those actual pieces you use, you can end up with very many different kinds of final outcomes. And so that's basically cartooned here. We already talked about skipping exons just because there's no SR protein binding here. Turns out you can have different splicing sites inside your exons, and that's what's shown here. In a few cases, you have, and these are just examples here. I don't expect you to remember all of them, but just, but that you can have splicing that happens basically anywhere. And it's going to depend on where you have the U2 binding to your particular splice site. You even in some cases have exons which can be used in one place and can't be used in other cases. Where you start makes a difference. Where you make poly A tails is going to make a difference. And we'll look at some of those examples here in the last couple of minutes of lecture. So one example where, in fact, a number of diseases are known is where you have skipping of exons or different splice sites that actually get used. In normal beta globin, you've got these 3 exons, there's only 3 of exons, where you have normal splicing that takes place. If, however, you have a mutation that happens to be right at the beginning of one of these exons, this is known in a number of patients that have trouble with their beta globin, you'll have a single nucleotide change here. The single nucleotide change means that U2 is not going to bind there anymore because U2 is always interacting through base pair interactions. You have a change in the base pair here. Now you skip this exon completely. You can have a single nucleotide change that now allows U2 to bind there, which otherwise wouldn't, which will give you a different exon here. And in some cases, it starts out with a single nucleotide change, which will give you a putative binding site now for U1, so you can start here. All 3 of these are actually quite well-known changes in globin that you see in beta-thalassemia patients. And a lot of the times when we have single nucleotide changes which lead to some kind of disease, it's almost always in a splicing change. Because splicing has so few nice consensus sequences, you can splice all over the place. It's great because you can mix and match these different exons in order to get one gene that can function slightly differently in a whole bunch of different cell types. But it's also a little dangerous if you do have, for instance, DNA damage that doesn't get repaired properly. You can end up with lots of changes in splicing. And I've actually had 2 students work in my lab, in fact, that have had these kinds of genetic changes. One had some changes in splicing in his collagen gene. Another one had, in fact, this exact beta-thalassemia that was present here. So we'll stop here. We'll talk about self-splicing introns next time.